place.
The scientists had triggered the changes by damaging the fly's DNA. Within each cell of the developing embryo is a chain-like molecule called DNA. The experiment showed that DNA was somehow causing the embryo to divide into segments. But how? Scientists were just beginning to grasp that the DNA itself was made up of segments, called genes. The question was, how did the genes shape the body? One researcher, Dr. Ed Lewis of Caltech, studied this question for 30 years by crossbreeding thousands of flies. Lewis's work led him to a controversial idea. He proposed that a surprisingly simple mechanism was shaping embryos. He wrote that each segment of the fly was being directed to grow by a single gene. A small set of genes, a kind of genetic toolkit, appeared to be laying out the entire body.
And as you looked at these genes, you said, this one affects this part of the body, this affects the next part of the body, and this affects the next part of the body. That was an astonishing observation.
It was astonishing because it seemed too simple. Nobody else thought single genes were powerful enough to control something as complex as the structure of the body. Skeptics argued that Lewis's idea was guesswork. Of course, he had never seen the genes, because the techniques to do so didn't exist.
From the 1920s to the 1970s, it was not possible to physically isolate any specific gene. That opportunity first became available, fortunately for me, at the time that I was a student. And so many of us thought, wow, we can finally dig in there and identify these really mysterious genes.
Levine enlisted his friend and fellow scientist, Bill McGinnis. The first gene they went after had an unusual name, antennapedia, which means antenna leg. The gene was thought to control the growth of legs. When the gene misfired, flies grew legs in the wrong place, on their heads in place of antennae. In normal flies, legs grow from the midsection, the area called the thorax. So Levine and McGinnis decided to hunt for the gene in the thorax of a normal embryo.
The expectation is that antennapedia would be active, expressed in the thorax, the developing thorax of the embryo. But who knew?
Levine and McGinnis had to do something no one had ever done before. They had to find a way to see a gene in action.
We wanted to light up the gene. And it was very painstaking work.
The project called for new and untested methods.
At first, it didn't work very well, and there were a number of technical problems to solve.
The team had to find a delicate balance of radioactive probes and toxic enzymes. Too much of either would destroy the embryos.
The process was not very gratifying on a day-by-day basis. Unbelievably tedious.
It took months of trial and error.
People often said, you know, you should try something else. You know, this is too long shot. You know, you're just wasting your time. But we kept going.
Finally, late one night,